Hello, everyone. Welcome to today's live broadcast, Antha, an operating system for seamlessly linking experimental design and lab automation. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by PerkinElmer. PerkinElmer is a global leader focused on improving the health and safety of people and the environment. Its dedicated team of 7,700 employees worldwide are passionate about providing customers with an unmatched experience as they help solve critical issues in human and environmental health. The company's innovative detection, imaging, informatics, and service capabilities, combined with deep market knowledge and expertise, helps customers gain greater insight into their science to better protect our environment, our food supply, and the health of our families. For more information, please visit www.perkinelmer.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Sean Ward, founder and Chief Technology Officer, Synthase Limited. Sean is a serial entrepreneur, first in the music industry with Relatable, and currently in synthetic biology with Synthase, where he is the architect and driving force behind Antha, a high-level language and operating system for working with biology. Before founding Synthase, Sean was a research associate in bioinformatics at University College London, where he conducted research into protein folding, protein structure prediction, and gene coding. At Relatable, Sean invented audio fingerprinting, which was used by major peer-to-peer companies such as Napster. Sean is also an advisor on synthetic biology to the Biotechnology and Biological Sciences Research Council and a SynBio Leap Fellow. I will now turn it over to Sean Ward for his presentation.
Thank you, Judy. As she mentioned, I'm a synthetic biologist, which means I take a slightly different perspective about how to understand biology. Mostly it means that I cheat because I get to make changes to an organism and try to understand what that change does by empirically validating through a variety of different assay protocols and a different number of technologies what it actually does. And that's key because, let's be frank, working with biology is hard. At its root, working with biotechnology takes too long, costs too much, and fails too often. And that's a problem that faces every single application that we have with biology, be it how we can develop a new drug, how we can develop a new industrial biotechnology manufacturing process, or how you can bring new crop traits to market in ag science. At the heart of it is the fact that, of course, you're dealing with complexity. I mean, the number of interacting factors that you encounter when you start talking about writing a genome is exponential, not to mention all the other interactions that take place with the environment, with the feedstock.
We have a brief technological difficulty. Stay with us. We will repair it immediately, and Sean Ward will be right back with us. So just bear with us for just a moment.
Apologies, we had a minor technical difficulty there. Are you able to hear me again?
Yes, we are.
Okay, excellent. So we just discovered the third problem in action, which is that even complex systems like phones have traceability issues at times. The heart is that everything in biology is context dependent. if you transplant a gene, say for the production of an industrial enzyme from one organism to another, you're also transplanting what is the entire machinery used to both produce, to fold, and potentially post-translationally modify that protein. And it may completely alter the toxicity, the function, and even the fundamental ability to have any useful product from that action. Now, unpicking these sorts of high-order interactions that exist between the context dependence of biological function is hard, even in the best of times. But the problem is that we compound that with the problem that our base working practices themselves don't give us definitive understandings of what's going on. PLOS Biology did quite a damning report back in January when looking at the fundamental research practices, call it the working practices with which we work with biology and with which we are ultimately trying to infer our conclusions, were themselves grounded in sand. Out of 441 randomly selected publications, only one of them was actually even theoretically reproducible by having enough information contained within the methods section that a third party could do that work. What it fundamentally is showing is that in biology, we have a real problem with the transfer of tacit knowledge, the ability to effectively describe how it is that we actually do our work. And without the ability to describe how we work, well, the ability to understand whether or not any particular result was a product of that mechanism of collecting the information about it, or whether or not it was actually a result of the biology that you were investigating, is an extremely unclear situation. And that's why major reproducibility studies in cancer therapeutics by Bayer and Amgen have shown that 25% and 11% of actual beliefs about what's going on in cancer systems were actually reproducible by third parties. Which, again, there's little wonder why that's the case when our basic lab practices themselves will vary from person to person within the same lab, let alone between different institutions. Ultimately, what that means is that we're trying to work on incredibly complex systems when we don't even necessarily know whether the outcomes that we're looking at were coming from the biology or simply the practices with which we're engaging with it. In fact, it's why when you look at the productivity, our ability to effectively work with biology, there's a negative exponential that's gone back for the last 65 years. In fact, it has a name, Eroom's Law. And no other field operates with this sort of negative productivity effect across the years. Now, what that means is that inflation adjusted, every 18 months it doubles in the cost to bring a drug to market. Now, that is, of course, a confounded problem with regulation, and, of course, the fact that you might say many of the low-hanging results in biology in terms of drug indications have already been collected. But you encounter similar challenges when working with biology in other contexts. In industrial biotechnology, it still takes, on average, 7 years to bring an industrial process to the commercial scale, and north of $100 million worth of investment. And in ag science, you're talking about over a decade, and on average, $300 million to bring a crop trait to market. In both of those cases, it's the inherent complexity of the biological systems that we're dealing with that are our fundamental limitations. Now, when people think about dealing with complex systems and exponentials, the most classic one is Moore's Law from the semiconductor industry. Essentially, that's the fact that the number of transistors we can put into a processor has doubled every 18 months. As you said, the inversion of what has been the productivity curve in the pharma industry. Now, that's an entire stair-step of various innovations that's led to it. But it was a change that happened in the early 1980s. The introduction of electronic design automation, and in particular high-level languages, VHDL and Verilog, which was the single most important transformation in the course of semiconductors. It meant that all of a sudden, the still only 30,000 semiconductor designers that were on the planet could have all of a sudden an entire tool stack with which they could engage this exponentially increasing problem of how do you design a chip where you're going to have to deal with twice the number of moving pieces as you did in the previous 18 months. And it also meant that that tool stack could transition the way that we fundamentally worked with semiconductors from, in the early 1960s, our first planar integrated circuits that were entirely designed on a piece of paper, designed with a human mind, laid out by hand, and ultimately fabbed by hand, to the modern processes today at a place like Intel, where billions of transistors are designed around, using an entire tool stack that, in the parlance of the industry, compiles the silicon, because these design tools talk directly to the hardware that ultimately is creating the physical objects that ultimately become these products. So, recognizing that we needed to be able to start to move up this ladder, to have a tool stack that allows us to engage with the complexities of working with biology, at Synthase we created Antha, which is very much like VHDL and Verilog, a language for describing what you are physically trying to produce. What is the experiment that you're trying to conduct? And it's the start of what we hope will be the entire design tool stack that ultimately will let us address grand challenges like, how do you engineer an entire genome? How do we more cost-effectively produce drugs? How do we have biomanufacturing processes that help wean us off of oil? Now, why are languages so important for productivity? The frank reality is because they're a way to encapsulate and manage complexity. The average professional computer programmer still only produces 10,000 lines of fully debugged, fully documented code per year. That's remained the same since about the 1960s. However, clearly, what we do with information technology has progressed rapidly since then. You can build, an iPhone app visually in the course of days today. And that's because instead of describing all the low-level details associated with, programming instruments, telling it how to move bits around in registers of a processor, like you do in assembler, we instead have high-level languages like Python, which allow us to express the intent behind our actions, such as that we want to print "Hello World" to the screen. Compilers are also an important piece of this tool stack. They're what helps translate a language and check the semantics. It's how you can do the equivalent of a grammar check and a spell check atop what you have written within a language. And it helps to catch many of the mistakes that can be caught automatically, and so empowers a user to be able to be more effective, to help debug the thought processes that are represented within their language. So in the same way, Antha exists as two languages, one for describing workflows as a composition of elements. These elements themselves are then defined textually as reusable and executable working practices. Think things like the way you can stitch DNA together, or transformation protocols, or assay procedures. In turn, these definitions of what you are trying to conduct as a biological experiment are made executable by the Antha operating system. This talks to various pieces of laboratory equipment through drivers and drives the physical execution of your experiment. And importantly, it also deals with translation between environments. So the same protocol can be written for, as an example, a Gilson Pipette Max, or for a PerkinElmer Janus G3. It also deals with many of the logistical problems that you encounter when planning, designing, and executing an experiment. In many respects, this is like enterprise resource planning software brought forward into the laboratory, dealing with your problems around scheduling, about whether or not you have the right reagents to be able to conduct this experiment, and integrating all of the information outputs that come from your experiment. Now, let's get a little bit more concrete about what we're talking about here for elements, and why systematically looking at your basic working practices is so important. So, as a synthetic biologist, if I can't make the DNA constructs, the interventions into the genome of an organism with which I'm trying to work, well, I'm kind of sunk. It's the bread and butter that allows me to unlock the entire range of genetic interventions that I'm trying to engineer to ultimately understand their function or effect on an organism. In our case, we use a variant of an assembly protocol based on type 2S remote cutting restriction enzymes. What that essentially means is that the recognition site is distant from where the cut is made within the DNA. And that means that you can essentially define a pure series of overhangs so that after the cut is made, the recognition site is no longer within the region that has been cut out. This means that you're able to very quickly assemble complex constructs where the different component pieces within your entire construct can be modularly swapped around or replaced by alternates within any particular slot. These remote cuts then can, in a single pot reaction, be mixed together with the ligase, and because you have these overlaps that are compatible, they will only assemble in one direction. The net result of this is that you have a very simple protocol for being able to assemble potentially combinatorial numbers of parts. The other piece, though, is you can start to look at a practice like this and investigate what it is that makes it successful. In our case, like many originally academically derived technologies, it would work about half the time, yielding a proper colony. I mean, anyone who's worked in a wet lab has had that soul-shaking experience of walking in to check your plate and finding out that there's nothing on it. So you definitely want that to be a high figure. But unfortunately, it wasn't necessarily going to be a good sign just to have a colonies, because half the time you would end up with the wrong construct as well. However, it turns out that if you systematically investigate all of the low-level details of a working practice, like, say, stitching together DNA, it's possible to find a part of the design space, optimizing things like which enzyme you're utilizing, your concentrations of DNA, the temperature that you run it with, and whether or not many of the standard practices that we take for granted in the laboratory, like, as an example, that you may have to heat shock your cell lines to do the transformation, end up being wrong. And finding the right way of running this means that you can build a protocol that works all of a sudden 100% of the time with colonies, and 99% of them are the correct construct. So you've transformed yourself from a position where your lab practice was an unreliable and artisanal operation, where you were bottlenecked heavily by tracing down your constructs that didn't assemble, to it being something that could be performed by any person and allows you to now build thousands of genetic interventions at once. And having done so, you can now record it in Antha. Now, as I mentioned, Antha is a language for describing these executable working practices. So in this case, construct assembly consists of saying that you are going to mix into an output plate a series of samples, including genetic parts and enzymes, and that you're following that by incubating at 2 different temperatures. This is something that, even if you perhaps haven't written code before, you should be able to read and understand. But more importantly, by representing a working practice in this form, you have something that can be translated into a variety of environments. In this case, that ultimately means various liquid handlers. Also, this systematic structuring and thinking through about what parts of a protocol were actually important means that you can go from much more complicated protocols, such as Gibson assembly or even Golden Gate, which require the use of PCR as part of the amplification process, that don't allow part reuse because they require things like a specific primer for an assembly, that require complex integrated work cells that have things on deck like, say, bead purification, or that require thermocycling, and that ultimately means you can have a much simpler and more robust lab process and protocol, which means that you can translate for comparable execution into a whole variety of manufacturers' environments, including hardware from SciBio, Gilson, and I'm happy to announce PerkinElmer with their Janus platform. Now, this sort of protocol optimization isn't limited to simplifying something like construct assembly. It's just as important to be able to systematically investigate what are the factors that are driving the successful execution of assay protocols. So, as an example, an experimental design such as a definitive screen can be used to simultaneously improve the signal-to-noise and also understand what are the major drivers of the success rate of something like an immunoassay. And to be clear, the signal-to-noise on your assay is the fundamental bottleneck on your ability to draw insight and ultimately definitive conclusions about what's going on in a complex biological system. And of course, if you don't model the complexity that drives the things like the successful signal-to-noise of an assay, you may find that instead of being able to have optimized it, you're sitting right on an edge, and you can't even truthfully trust the outcomes that came from your assay. And as I mentioned, the nice thing about having written it this way is that it will translate from extremely capable instruments that can run complex integrated processes on themselves, such as the Janus, all the way through to simple actions like a Gilson pipette max or even manual workflows. So where do we see are the changes that have to be made in how we work with biology? For us, it starts by a robust characterization of our inherent lab practices. If you don't have reliable working practices, if you can't transform your cell lines, if you can't effectively make genetic interventions, if your assays have poor signal to noise, well, you basically cannot draw definitive conclusions. Also, if your fundamental lab practices are unsound, automation is a problem. It's not an asset. If only 25% of your constructs are successfully assembled and you now try to make thousands using automation, well, then you've just created hundreds of problems for yourself. On the other hand, if your fundamental working practices are reliable and robust, automation is a force multiplier. It means that all of a sudden you have the potential to flexibly execute complex experiments that give you a sufficient experimental bandwidth that you can properly apply multifactorial experimentation to unpick definitively what's going on in a biological system. And of course, being able to build successful models about what is going on in these biological systems means that you can properly start to engineer with biology, which is a critical bottleneck in our ability to apply it in areas such as industrial biotechnology. To be clear, I'm not speaking about this just from principles. I'm speaking about the lessons that we've already learned at Synthase. This is the transformation that we've made ourselves, and which Antha and our Antha operating system was the linchpin to being able to make this transformation. So how does all this work? Well, as I mentioned, at the top, it's about having a robust model of what is the experiment you're trying to conduct. So Antha visually allows you to define the flow of information and physical objects between these elements. So taking a standard sort of synthetic biology workflow, that means things like planning what construct you're trying to make, checking whether or not you have to synthesize DNA or retrieve parts out of your freezer. It then means stitching together that DNA, transforming a cell line, growing it up, and then validating that you've successfully made the correct constructs. The elements themselves are then a robust way of describing any one of those unit operations. This is a programming language which is based on an extension of the Go language from Google. So you can do any computational workflow you want, including complex bioinformatics pipelines or complex modeling tools right within these elements. But it importantly extends it into describing actions in the physical world. So Antha elements start by defining what do they depend on. This can be, as an example, specific pieces of equipment, such as liquid handling, or different pieces of analytic kit, such as a plate reader. This can also be a robust definition of what is the specific other working practice that this depends upon, such as the repository URL on GitHub of another Antha element. There's no, in Antha, searching for the methods section of a paper from 1976. Next major section is the parameters and data, which defines all of the information flow in and out of an Antha element. This has a strong type system, so Antha understands the difference between a volume and a temperature, and whether or not you're expressing microliters or liters or picoliters. This is important because this is the interface for this working practice. In essence, it's what are the variables that you may need to optimize. Inputs and outputs is similarly a type system over the physical objects that are coming in and are produced by an element. So it knows the difference between a genetic construct and a cell line, and thus is able to tell you what working practices can be compatibly linked together. The requirements section allows you to do input testing. So something like a construct assembly protocol is often going to be dependent upon the types of inputs that you're running. So for example, you can make sure that you don't have within the coding region that you're trying to stitch together any restriction enzyme sites that are going to be in conflict with the restriction enzymes that are utilized in that construct assembly protocol. Now, the heart of an Antha element is the steps block, where you're describing what you do to in the context of a single sample going through the system. Now, that's because the complexity of describing how you manage thousands of simultaneous threads of an experiment is hard for professional programmers. And it's an incredibly difficult problem to schedule and map it all down to things like individual plates and when they are being moved. As such, you always write from the standpoint of a single thread of execution, a single sample, and you have no information or access to information about the other samples that are being processed. However, there's certain classes of protocols, like an assay, where you do have dependence on other samples that are being used in the system, such as needing to be able to describe that there's a control curve attached with an assay. The setup block and then ultimately the analysis block enables you to pierce that veil and define these dependencies in a very structured way. So the system has the degrees of freedom it needs to ultimately optimize the execution of your experiment, and you have a simplicity of expression that keeps what you have to use to describe ultimately the complex execution of thousands of experiments at once as a simple process. Lastly, the validation section enables you to validate that you've actually executed this element successfully. Unfortunately, when we're doing an experiment in biology, it's often extremely expensive. And so catching that we've made a mistake early is key to being able to reduce that downstream cost. Now, unfortunately, again in biology, often finding out what is the current state of a biological system is destructive. And so you can tag tests that are either destructive or non-destructive. It can be optionally performed while you're conducting these experiments so that you have inline QC as part of your experimental workflow. A good example of a non-destructive test would be something like looking at the R-squared value of your control curve on your assay to determine whether or not it appears to have been executed correctly. So when it comes to actually working with Antha, it starts with sample management. If you don't know what all the physical objects are in your lab, what containers they're stored within, and what is the provenance of everything within those containers, and, of course, where those things are physically located, you can't, of course, properly describe the flow of samples and the flow of information through a workflow. The next major step about working with Antha is to define what's the experimental workflow that you want to run. Of course, that's easiest to do by showing you.
Before you can run an experiment, it's important to actually define what experiment you're planning to conduct. Antha provides repositories of your elements and workflows that can be shared both on an individual or organizational basis. Editing a workflow is completely visual, and you're able to drag and drop new elements out of your libraries into it. You can see here that every input and output of an element is visually coded as information flow or physical object flow, and can be wired directly up between elements. You also see a collection of unfulfilled inputs passing to the left. These are what are needed to actually launch a workflow. You can, of course, dive right into the code level of an individual element. So if you decide you need to make a change in a working practice, that can all be done completely in your web browser while defining this. Saving elements is all version controlled. Under the hood, this is based on Git.
So having defined a workflow, to be able to actually execute it successfully then, you need to pass in what are all the parameters that have been defined as empty within the workflow. And again, that means you can build visual tools to help you be assisted in planning, reasoning about, and executing your experiment. Again, the easiest way is to show that.
As mentioned previously, to run an Antha workflow, you need to satisfy all of the inputs. In many cases, this can be done by importing an input file from another design tool. In this case, taking a GDX file from DNA2's Gene Designer, which contains a summary of a number of constructs that need to be assembled. Antha is able to parse these inputs and give you an estimate of what's going to come out of it. It then is able to simulate the entire workflow, including all the low-level details, for the connected group of hardware that you're now requesting this workflow to be run against. This includes being able to tell you exactly how long this is going to take, and because it ties into the inventory system, how much is this going to cost. When you actually go into the details, you can find that all the low-level questions like what's the plate layout, what is the number of plates you're going to need, what are the consumables that you're going to need, how much do you need to make up of all of the different inputs for your workflow have been calculated for you. And again, because of the tie into the inventory management system, it knows the barcodes of the specific objects if they already exist in, say, your freezer. Having set up your deck appropriately following the instructions that have been provided by Antha, you can essentially go from a design of what you need to pass into a workflow to a robot starting to actually run your experiment in what is a grand total of about 45 seconds.
Now, ideally, you end up having an integrated work cell like a Janus, but the reality is that many people also have manual operations that they have to perform as part of their workflow. So Antha is designed to deal with hybrid workflows where you may have to physically intervene at different stages of executing your experiment. As such, we have an integrated timeline of what's taking place on all the different pieces of equipment and can assist all the different transfers that have to take place in the course of it. Again, showing is the easiest way.
Antha isn't just about running completely integrated work cells, however. The vast majority of people conduct experiments where they have to take manual actions as well. So Antha is designed to deal with multi-device, multi-element complex workflows where you do have to physically transfer objects between different pieces of equipment. Antha is able to notify you both in the web browser or via text messaging when it's time to take an action.
The net result of all of that, though, is that you've got a robust log of everything that's been conducted in this experiment, including the physical objects, all the information flow, what specific working practice was performed at every one of these stages, and ultimately a robust provenance of what was done to produce every piece of information in your experimental results. That rich data log is then something that you can readily pay attention, you can readily interpret using much more complex tools. Again, the easiest way is to show you that.
Antha produces a robust integrated log of everything that took place during the course of executing an experiment. It also provides a number of different ways to get access to the data that's contained within that log. Here you're seeing our Jupyter Notebooks integration, where you're able to completely in your web browser have a MATLAB-style analysis workbook sitting atop the data that's been collected within an Antha execution log. It's easy for an organization to develop these sorts of analysis workbooks. In this case, you're seeing one for multivariate design of experiment analysis, where you're able to automatically pay attention to all of the input parameters that you've done in an experiment and compare their potential effects against the various responses that have been collected out of the analysis elements that were integrated into this workflow.
Now, pulling all that off is quite a complex system under the hood. When you're actually working with Antha, these individual elements are compiling to standalone software binaries. That means that you have the flexibility of disseminating a working practice, be it a complex bioinformatics algorithm or just simply a better way of transforming, your bog standard E. coli cell line, either as a closed source piece of software or as open source. Obviously, I do prefer the open source model, but it is important to allow many different ways of working with this system to evolve. At runtime, there is a connected group of laboratory equipment run by our lab manager. This includes a scheduler dealing with all the low-level details of when do things move from one place to another. It also includes a sample tracking and sample management, paying attention to all the objects that are in flight at this single unit, such as a Janus G3. The workflow manager is responsible for actually firing up all the standalone elements that make up any arbitrary workflow that you're trying to execute against this connected group of hardware. And it also is able to, if necessary, launch elements up in the cloud because these are wired up over the network and do not have to be co-located on the same machine. The device manager itself ultimately is talking to your various pieces of lab equipment via our device drivers, which themselves are network services. So you can be talking to the laboratory control PCs that might be in a device, connected to a device in another room, assuming your IT department is willing to let you connect up to the network. And of course, all of the data and all of the events about what is actually producing these experiments and what's happening during the course of running them is being collected into the log service, which under the hood consists of both a NoSQL database, CouchDB, and also special time series record logs so that you can overlay information from things like environmental sensors, which is great forensics data when you're trying to figure out whether any particular data point is truly a black swan or just a result of experimental error. On more of an organizational basis, there's also a collection of cloud services, including user authentication, dashboard views for things like how many work cells an organization has access to, device management, so that you have an understanding of how many devices do you have, when were they last maintained, and of course organizational level experimental data repositories, as well as workflow repositories and element repositories. That means that within an organization you can share best practices, or for specific users, force them to use only validated practices. This is also where you can deal with executing much more complex workflows, such as being able to do things like a genome assembly pipeline being spun up in much more capable resourced, much more capably resourced cloud services. Everything on a user interface basis is designed to be run on touchscreen devices, including iPads and things like Windows 10 Surface devices, and is amenable for working with lab gloves still intact. Now, while that's fun technology, let's talk about the science that this actually lets you do. So at Synthase, as I mentioned, we engineer microorganisms. Most of the time for us, that's been about how do we use them for biomanufacturing. And the moment you start talking about all the complexity attached with a biomanufacturing process, it's into the hundreds of different interacting factors across environment, and of course with modern synthetic biology techniques, across the genetics as well. And it's impossible to deconvolve the two, because actually the outcome of the biomanufacturing process is a result of genetics and environment, not just the one. So how can you go ahead and unpick the high-order interactions that are going on in a system like this? Well, in our case, it's iterative multifactorial experimentation. So we'll start with a process that we call a factor screen, where you're casting a broad net. Now, this is an example from, for us, 4 years ago when we were still trying to do this stuff by hand. And believe me, you really don't want to be that guy. So even a very narrow question, like how to express an industrial enzyme, is going to require dozens of factors in truth, but in this case we did 15. The first question being, which of those 15 factors matter? And having gotten data, you're then able to identify, okay, we were looking in the wrong entire part of the design space. We need to move it. Having done that, we were then able to start to refine this, having identified that in fact many of those factors were having no effect. Luckily for us, biology is filled with these cases where there are many potential interactions, but the vast majority of them within the particular part of the problem space that you're looking at don't matter. That gives more experimental bandwidth to start to refine and unpick which of these interactions are truly mattering. And having identified what were the key factors driving the ultimate result yield of this particular industrial enzyme meant that we could finally unpick the true interaction that was driving this entire system. To be clear, this is fundamentally sitting atop building a model of what might be occurring in the biological system, determining what observations are going to tell us the most information, then using Antha to physically do the experiment, collect the results with biology telling us what's going on, and ultimately using that to refine our model. So in terms of actual data, it's not a huge number of experiments that are required to do this sort of iterative investigation into biology, but the complexity of every single observation is extremely high. So you're talking about, in the course, in this case, of improving the manufacturing yield of an industrial enzyme 200-fold in 4 weeks of lab time, with an additional lab week spent just to validate the model, sampling around it. To be clear, each of these different colors is the results of 1 week of laboratory execution. And unpicking what drove those changes, well, out of those first experiments that were just testing if an effect occurred, you immediately saw a two-factor interaction between chaperones and time. Chaperones being a genetic factor for what machinery was being transplanted into the organism to help proteins fold successfully. Unpicking that two-factor interaction, though, just is the start of your journey, because it turned out once we had moved to the right part of the design space, there was in fact a three-factor interaction going on in this biomanufacturing process between genetics and environment. And without high-dimensional integrated experimentation, there was no other way to have possibly unpicked this. And to be clear, getting these factors right was the difference between that 200-fold yield improvement and about half that in the average case, and if you got them wrong, being on the edge of what we call a design cliff, getting absolutely nothing out of your biomanufacturing process. These design cliffs, these sensitivities to parts of the environment or parts of genetic space are all over the place in biology and are one of the major drivers of why you'll have a laboratory practice that works one week and not the next. But you might say that there's a limit to how complex of a system you can deal with. You can obviously do this in E. coli, but not in much more complex problems like how do you produce a biotherapeutic. Well, more recent work we've been doing with Merck Pharmaceuticals, MSD, over here in the UK, has been on the development of a new biomanufacturing platform where, using Antha, we were able to conduct an experimental campaign on over 27 simultaneous factors. To be clear, that's looking simultaneously at genetic modifications to a cell line, changing what we're feeding it, and changing the environmental conditions under which it's being grown. And in the course of 10 weeks of laboratory time, we were able to unpick a 4-factor interaction between genetics and environment that was driving a 10-fold yield improvement in that production process. Now, because we're building models, however, we also had an unfair advantage. In a traditional process, you would have been making many variants, genetically speaking, and screening for hits, which means you would have gone with this colony right here. Unfortunately, as that organism starts to move up into a larger scale manufacturing environment, controlling temperature becomes extremely difficult. And you wouldn't have been able to unpick the fact that that particular organism was temperature sensitive until you put it into a large scale fermenter and watched your yield drop off. In contrast, by being able to conduct these complex experiments that could allow you to build models iteratively, you could unpick that by locking in with 2 different genetic attributes, you could build an organism that was robust against temperature variation and was still going to operate successfully once you move it into large-scale fermenters. This ability to do model building is fundamental to ultimately being able to do design for manufacturing and to be able to transfer the results of working with a biological system from one environment to another. It's the exact same principle that lets you transfer a working practice like construct assembly or a biomanufacturing process like making an antibody-conjugated drug. Now, in the context of just our simple biomanufacturing investigations, you still have quite a complex process, everything from factor mining to gene design to gene synthesis to construct assembly. And that itself then is all being driven by our experimental designs. Which factors, which genes are we going to insert into this pathway? Which genes are we going to knock down? All of that is ultimately used to build organisms that we're evaluating in a scaled-down environment, in our case, microwell, titer wells, where we're recognizing what parts of the transferred context, ultimately large-scale fermenters, are we able to understand and unpick at this scale. It then means rapidly moving into stirred tank reactors. In our case, it's a system from Sartorius called an Amber 250, which gives us a context-appropriate environment for understanding the physical interactions going on with our organisms. And in turn, all of the data coming out of these has to go through an entire bank of omics pipelines, everything from looking for various levels of analytes to sequencing to understand genetic drift that's occurring in these organisms. And of course, ultimately, it's moving up into stirred tank reactors, steel, and confirming that the models are successfully transferring between these different environments. At the heart of all of this is being able to execute these complex integrated experimental pipelines that talk to many different pieces of laboratory equipment and solve all the scheduling and logistics challenges across these experimental pipelines. That ultimately means that you can start to build numerical biology, models of what's going on in these complex systems. And of course, feed that back into your experimental designs. Now, looking into the future of what is going to become possible to do with biology, we're also happy to announce that we've started a broad-ranging collaboration with Microsoft. This includes bringing Antha to the Azure cloud service so that you'll be able to design, plan, and execute your experiments purely from your web browser without having to think about the IT requirements for it. It also, however, is bringing deep learning technologies to bear so that you can start to turn a variety of your currently qualitative data outputs, things like microscopy image sets, into quantitative ones, because the heart of being able to unpick these complex systems is ultimately being able to bring the right data out of them so that you can build informed models of what's going on with these systems. So in summary, Antha is, for us, a ground-up reimagining of how you engage with working with biology. It's about how you can talk to all the laboratory equipment. How can you cut through the bottlenecks that exist in your ability to plan, design, and execute the complex experiments it takes to ultimately gain complex understanding into systems such as biology. Antha is open source. You can get access to it right now on GitHub. And if you're finding out that this is something that's of interest to you, I recommend that you dive in at anthalang.org. Again, I want to thank PerkinElmer for this opportunity to present what we're doing with Antha and their support now for Antha by helping us bring Antha to their Janus G3 platform. And I want to turn it over to any questions you might have. Thank you.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. Try to answer as many questions as we can. Our first question is, what happens if you are modeling a biological system and miss a key factor in your model?
Thank you. So, an effective part about multivariate investigations is that you're fundamentally building a model of whether or not the variance that you're seeing in your data outputs, such as the assay of a product, is explained by the variables that you're taking a look at in your multivariate model. So you basically are looking, is there unexplained variance? As such, if you've missed a major factor, there's going to be a large amount of it. If you haven't, well, then luckily enough, then you're able to move ahead with a proper model of what's going on in that system. When you're running these complex campaigns also with hundreds of runs attached to them, your p-values associated with your conclusions are extremely high. So it gives you an ability to actually definitively state that the effect that you're observing is a real one and is explained by the variables that you have investigated in your experimental design.
Okay, we'll give people another moment to submit more questions. If there are no more questions, I would like to once again thank Sean Ward for his presentation. Do you have any final comments for us today?
We look forward to seeing what can happen as people are able to systematically codify, transfer their tacit knowledge, and characterize their fundamental working practices, which is going to give us a collective foundation with which we can truly unpick the complexities of biology.
Sean, a question has come in. I'd like to pose it to you. The person says, I see and understand the Antha concept from a Lean Six Sigma concept. The question is, how do we get Antha for streamlining Lean Six Sigma biology research?
Sure. So you're correct that there's quite a few principles of quality by design baked into Antha. In essence, it's trying to bring that all the way upstream into your early discovery work. So one way you can engage with is with the open source technology that's already available there. You can also engage with us directly, as we have all of the visual tools that you've seen presented in this presentation already available for access shortly in a cloud-hosted form, and also for enterprise licensing to be deployed inside of your own organization.
Well, thank you. I would also like to thank our sponsor, PerkinElmer, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through November 12th, 2016. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Bye.